Hi students, today I am going to deal the topic chromatography from the chapter mass spectrometry and chromatography. Today I am going to deal the topic chromatography from the chapter mass spectrometry and chromatography. Learning objectives. Upon completion of this presentation, you will be able to understand the concepts of absorption, adsorption, chromatography, classification of chromatography, gas chromatography, components of gas chromatography, construction and working principle of gas chromatography, advantages and limitations of gas chromatography, and applications of gas chromatography. Upon completion of this presentation, you will be able to understand absorption and adsorption, chromatography, classification of chromatography, gas chromatography, the components of gas chromatography, construction and working principle of gas chromatography, advantages and limitations of gas chromatography, and applications of gas chromatography. Absorption and adsorption. These two are the most important phenomena which are very useful in chromatography. Absorption. Absorption is a physical or chemical phenomena or a process in which atoms, molecules or ions enter some bulk phase like the liquid or solid material. It is the process in which a fluid is dissolved by a liquid or a solid. It is a process in which fluid is dissolved by a liquid or a solid, by a liquid or a solid absorbent. Absorption is the process or a phenomena. It is a physical or chemical phenomena or a process in which atoms or molecules or ions enter some bulk phase like liquid or solid material. It is the process in which a fluid is dissolved by a liquid or a solid, that is absorbent. The second phenomena is adsorption. Adsorption is the adhesion of atoms, ions or molecules from a gas, a liquid or dissolved solid to a surface. Adsorption is the adhesion of atoms, ions or molecules from a gas, liquid or dissolved solid to a surface. Adhesion of atoms, ions or molecules from a gas, liquid or dissolved solid to a surface. This process creates a film of the adsorbent on the surface of the absorbent. So adsorption is the adhesion of, it is sticking of atoms, adhesion to come closer together, adhesion of atoms, ions or molecules from a gas, liquid or dissolved solid to a surface. This process creates a film of the adsorbent on the surface of the adsorbent, The difference between both the phenomena, absorption and adsorption. The difference between the two phenomena, that is absorption and adsorption. What is adsorption? Adsorption is a surface phenomena. Adsorption is a surface phenomena. Whereas absorption, it concerns with the whole mass of the absorbent. It concerns with the whole mass of the adsorbent. Whereas this adsorption is only on the surface, it takes place only on the surface. And adsorption, in it, the substance is only retained on the surface and does not go into the bulk or interior of the solid or liquid. The substance is only retained on the surface and does not go into the bulk or interior of the solid or liquid. It doesn't go into the bulk or interior of the solid or liquid. In it, the substance is only retained on the surface and does not go into the bulk or interior of the solid or liquid. It implies that a substance is uniformly distributed. A substance is uniformly distributed through the body of the solid or the liquid in absorption. Then in adsorption, the concentration of the adsorbed molecule is always greater at the free phase. The concentration of the adsorbed molecule is always at a free phase. It is always greater at the free phase. Whereas in absorption, the concentration is low. Whereas in adsorption, the concentration is greater than at the free phase. Greater than at the free phase. Adsorption is rapid. It is rapid in the beginning and it slows down near the equilibrium. It is rapid in the beginning and slows down near the equilibrium. Whereas absorption, it occurs at the uniform rate. It occurs at the uniform rate. So adsorption is rapid in the beginning and it slows down near the equilibrium. Whereas absorption, it occurs at the uniform rate. It occurs at the uniform rate. Adsorption, it is rapid in the beginning and slows down near the equilibrium. Examples of adsorption are water vapor adsorbed by silica gel and ammonia is adsorbed by charcoal. Ammonia is adsorbed by charcoal. So water vapor adsorbed by silica gel and ammonia is adsorbed by charcoal. These are the examples of adsorption. Whereas in the case of absorption, water vapor absorbed by anhydrous calcium chloride. Water vapors absorbed by anhydrous calcium chloride. Ammonia is absorbed in the water forming ammonium hydroxide. Ammonia is absorbed in the water forming ammonium hydroxide. So this is an example of absorption. Water vapor absorbed by anhydrous calcium chloride. And ammonia is absorbed in the water forming ammonium hydroxide. NH4OH. So this is the difference between absorption and adsorption. Absorption and adsorption. What is chromatography? Chromatography is a technique. Chromatography is a technique. It is a Greek word actually. Chromatography is a Greek word. Chromato means color. Graphy means to write. So chromatography is a technique for separation of mixtures into their components in order to analyze, to identify, to purify and or quantify the mixture of components. Quantify the mixture or the components. So you have a mixture. The mixture contains several components in it. So what does chromatography do? It is a technique of separation of the components of the mixture. For what purpose? To analyze, to identify, to purify and or to quantify the mixture or the components. So you can clearly observe the picture. In the picture, the mixture is containing four types of components. Yellow colored balls are there, red color balls are there, blue color squares are there and black dots. These are the components of the mixture. So they can be separated by a process called chromatography. So all the four components are separated. You can see all the yellow balls, 3 balls are formed as one group. Or the 4 red balls are formed as another group. And the 3 blue squares are formed as one group. And the 2 black balls or circles are formed as one group. So you are separating the component of the sample by the process, by a technique or by a process called chromatography. Chromo means color, to write, to separate two different components. So for what purpose you are doing? To analyze, to identify, to purify or to quantify the mixture or the components. These are the four things which we are going to do. Either you analyze the components, or you identify the components, or purify the components, or quantify the components. So chromatography is used by scientists. It is used by scientists to analyze. Analyzing means examine a mixture, its components and their relationship to one another. Analyzing means examining a mixture, its components, what are the components present in that mixture, and their relation to one another, and their relations to one another. That is called analyzing. So scientists use this technique, use this analytical tool to analyze, to examine a mixture and its components and their relationship with one another. Then to identify, determine the identity of a mixture or a component based on known components. To identify is to determine the identity of a mixture or components based on known components. Then purify, separate components in order to isolate one of the interest for further studies. So you want to separate the particular components in order to isolate one among them for our interest of study. Then quantify, determine the amount of the mixture and or the components present in the sample. You are going to determine how much quantity or how much amount that component is present in the sample. So these are the four things or four processes which a scientist or which a person, when the sample is subjected to chromatography, he can do these four things. Either he can analyze the sample, or identify the sample, or purify the sample, or quantify the sample, So chromatography is defined as a physical method of separation of the components of a mixture by distribution between two phases. There will be two phases. One is called the stationary phase and other one is the mobile phase. One is the stationary phase of large area and other one is the mobile phase which passes along the stationary phase. So definition of chromatography: it is a physical method of separation of the components of a mixture by distribution between two phases. One is the stationary phase of large area and second one is the mobile phase which passes along the stationary phase. What are the advantages of chromatography over other methods? There are several other separation methods of separation of components of the mixture, but why there are certain advantages of chromatography over other methods? Like, they are useful for separation of mixtures of chemicals having closer physical or chemical properties. It may be difficult to identify both of them or separate two components almost similar in their physical and chemical properties. So that is possible with chromatography, and it is a gentle method, and separation in even micrograms is also possible using this method, and it is a simple and rapid and efficient technique. There are other methods of separation of components of the mixture, but the advantages of chromatography over other methods is Separation of mixture of chemicals having closer physical or chemical properties can be achieved with this method. And this is a gentle method, and the separation in even micrograms is also possible very easily, and it is a simple and rapid and efficient technique. It is a simple, rapid and efficient technique. Classification of chromatography. Classification of chromatography. So chromatography can be classified as gas chromatography and liquid chromatography. Basically, they are classified into two types, gas chromatography and liquid chromatography. In both the chromatographies, they have two phases. One is the stationary phase and other one is the mobile phase. In the gas chromatography, the mobile phase is in the gaseous state, whereas in the liquid chromatography, the mobile phase is in the liquid phase. That is the difference between gas and liquid chromatography. So, the basic classification of chromatography are, we have gas chromatography and liquid chromatography. Gas chromatography and liquid chromatography. Gas chromatography where the mobile phase is in gaseous state, that is said to be gas chromatography. We have in the gas chromatography two sub-classifications are there, gas and liquid and gas and solid. The stationary phase can be either liquid or solid. So, gas is the mobile phase and liquid is the stationary phase. Gas is the mobile phase and solid is the stationary phase. These two are the methods, are the types of gas chromatography. When we come to liquid chromatography, we have three types. Liquid chromatography is paper liquid chromatography, column type chromatography and thin film type. Paper type liquid chromatography, also in shortcut we can write as LC, paper type liquid chromatography. Column type, in column type we have liquid solid, gel permeation and ion exchange. Liquid solid, gel permeation and ion exchange. And we have the next one is your thin film type. So, chromatography is classified into two basic types, gas chromatography and liquid chromatography. In gas chromatography, we have two types, gas and liquid and gas and solid. In liquid chromatography, we have three types, paper type, column type. In column type again we have four types, liquid solid and gel permeation and ion exchange. And the last one is thin film type. So, I am going to deal the topic gas chromatography today. The principle of gas chromatography is a type of chromatography in which the mobile phase is a carrier gas. As I told you, chromatography we have two types, two phases. One is the stationary phase and other one is the mobile phase. In gas chromatography, the mobile phase is a carrier gas. So, usually an inert gas such as helium or unreactive gas like nitrogen is used as a carrier gas. So, the mobile phase is a carrier gas. Usually an inert gas such as helium or unreactive gas like nitrogen is used. The stationary phase is a microscopic layer of liquid or polymer of an inert solid support. Liquid or polymer of an inert solid support. As I told you, gas chromatography is of two types, gas liquid and gas solid. So, the stationary phase is either a liquid or a polymer of an inert solid. So, you can have gas liquid chromatography and gas solid chromatography. The instrument performs the gas chromatography process. So, the components of gas chromatography, the basic parts of a gas chromatography are, you have the carrier gas supply along with the pressure regulator and flow monitor we have the sample injection system, chromatographic column, thermal compartment or thermostat, detection system and strip chart recorder. I'll show you the figure in the following slides of the gas chromatograph. The basic parts of gas chromatograph are carrier gas supply along with pressure regulator and flow monitor. Then sample injection system. Then we have chromatographic column. Then thermal compartment or thermostat. Then we have the detection system, and the last one is the strip chart recorder. Strip chart recorder. Gas chromatograph is different from other forms of chromatography. Gas chromatograph is different from other forms of chromatography. In this, the solution travels through the column in a gas state. It is different from other forms of chromatography. In this, the solution, it travels through the column in a gas state. It interacts with the walls of the column. It causes different compounds to elute at different times called retention time. The comparison of this retention times is an analytical power to GC, that is gas chromatograph. So, it consists of carrier gas supply, then your sample injection system, then your chromatographic column, then the thermal compartment, and we have the detection system and recorder system. So, in gas chromatography, it is different from other forms of chromatography. In this solution travels through the column in the gaseous state. That is, the sample which you are using for analysis is traveling through the mobile phase is in the gaseous state. So, it interacts with the walls of the column. It causes different compounds to elute at different times called retention time. The comparison of these retention times is the analytical power to the gas chromatograph. So, this is the structure or the block diagram of or the schematic diagram of your gas chromatograph. As I told you, you have a cylinder there. The cylinder holds the carrier gas. Then we have the carrier gas supply, it is called. Then we have pressure regulator and flow regulator to control the flow rate and pressure values of the carrier gas. Then we have sample injection system. The sample is injected into the column, chromatographic column. The chromatographic column is temperature controlled using an oven. So you have a thermostatic operation there to control the temperature of the entire column. So, the chromatographic column is the heart of the chromatograph, and the output is taken from the end of the chromatographic column based upon the elution of the products of the components. Each component has its own diffusion or its own retention time, the time to retain itself in the column, and it will be eluted based upon the time of based upon the type of retention, and you will be a plot of the retention time and a graph is drawn using a recorder. Okay, so this is your basic block diagram of chromatograph. So the first part is the carrier gas supply. Carrier gas in a high pressure cylinder with attendant pressure regulators and flow meters. So helium and nitrogen or hydrogen or argon are used as carrier gases. So helium is preferred for thermal conductivity detectors because of its high thermal conductivity relative to that of the most other organic vapors. Mostly the other vapors, the carrier gas we prefer helium is because it can be used for thermal conductivity detectors. Nitrogen is preferable when a large consumption of carrier gas is employed. So carrier gas from the tank passes through a toggle valve, a flow meter. You have a valve, a flow meter with 1000 ml per minute and a capillary restrictors and a pressure gauge of 1 to 4 atmospheres. So pressure is maintained between that level, and also the flow rate of the carrier gas is also maintained between the range of 1 to 1000 ml per minute. Flow rate is adjusted by a needle valve mounted on the base of the flow meter, and it is controlled by the capillary restrictors. We are going to use capillary restrictors to control the flow rate of the carrier gas. The operating efficiency of the gas chromatography is directly dependent on the maintenance of constant gas flow. So, by using this regulator flow level, flow control and pressure control, you can maintain the flow rate of the carrier gas at a constant value. Okay, so this is about your carrier gas supply. Then the sample injection system. Liquid samples are injected by a you are going to use a micro syringe as seen in the figure earlier. You have a micro syringe with a needle inserted through a self-scaling silicone rubber septum into a heated metal block by a resistance heater. So gaseous samples are injected by a gas tight syringe or through a bypass loop and valves. Typically sample volumes range from 0.1 to 0.2 ml. Very small values of the sample is taken. So this is one of the advantage. You don't need to take large amount of sample. The quantity of the sample is very less, 0.1 to 0.2 ml. So this is your sample injection system. Then the third one is your chromatographic column. Chromatographic column, the separation column. The heart of the gas chromatography is the column, which is made of metals bent in U-shape or coiled into an open spiral or a flat pancake shape. So the heart of the gas chromatography is a separation column or the chromatographic column, which is made of metal bent in U-shaped or coiled into an open spiral or a flat pancake shape. Copper is useful up to 250 degrees centigrade. We have switch lock fittings which make column insertion easy. Several sizes of columns are used depending upon our requirement. Depending upon the requirement, you are going to use several sizes of columns. So this is the heart of the gas chromatography. Then your detectors and recorders. Detectors sense the arrival of the separated components and provide a signal. These are either concentration dependent or mass dependent. They may be either concentration dependent or mass dependent. So the detectors and recorders, detectors sense the arrival of the separated components and provide a signal. These are either concentration dependent or mass dependent. The detector should be close to the column exit and the correct temperature to prevent decomposition. So detectors should be very close to the column exit, and the temperature should be correct to prevent decomposition of the sample. Then recorder. The recorder should be generally 10 millivolts full scale, fitted with a fast response pen 1 second or less. The recorder should be connected with a series of good quality resistors connected across the input to attenuate the large signals. To attenuate the large signals, they must be connected with a series of good quality resistors. An integrator may be a good addition also. You need to have an integrator to make your signal much more available for recording. Then the operation of a gas chromatograph. Normally nitrogen or argon or helium is used as a carrier gas. A variable in compressed form is placed in a cylinder. It is fitted with a suitable pressure. The carrier gas is compressed in the compressed form, is placed inside the cylinder, and it is fitted at a suitable temperature. Now it is conducted from the cylinder through a flow regulator into a sample injection port. It is the sample injection port. Then after the sample is injected into the sample injection port, the chromatographic column, the next part is your column. It is maintained at a certain temperature T1, which is maintained, the sample is maintained at a temperature T1. It ensures rapid vaporization but not thermal degradation of the solute gas, and liquid samples are injected by the syringe. The solute vapor mixes almost instantaneously with the flowing carrier gas. It is swept into the chromatographic column. So, one side from the sample injection system, the sample in its vapor phase will enter into the column along with the flow of carrier gas. So, it is swept into the chromatographic column. So, it is the heart of the chromatograph. It is the heart of the chromatograph. The different solutes in vaporized sample are separated from each other by virtue of the different interaction with the column packing. The column is packed with a specific material. So, depending upon the retention time and the nature of the components of the sample will be passed on through the column. The column is maintained at a temperature T2. It determines the time for the passage of the solute. The resolution and efficiency obtained with the particular column. At the end of the column, the solutes emerge individually and they enter the detector. So, the detector produces an electrical signal corresponding to the quantity of the solute leaving the column. This signal is supplied to a potentiometric recorder. So, you get a plot. That plot is called a chromatogram. It is a plot of time and signal amplitude is obtained. Time signal amplitude is obtained. This will give the identity of the components in the mixture and the concentration. It gives the identity of the components in the mixture and the concentration. So, we have detectors. The most commonly used detectors in gas chromatography are helium ionization detectors, nitrogen phosphorus detectors, mass selective detectors, photon ionization detectors, pulse discharge detectors. These are the commonly used detectors: helium ionization detectors, nitrogen phosphorus detectors, mass selective detectors, and then mass selective detectors, photon ionization detectors, and pulse discharge detectors. At the end of the column, what happens at the end of the column? You will be placing a detector. So, the detector senses the arrival of the separated components and it provides a signal. And this signal is recorded, and that recorded output is called as chromatogram. It is a visual output of the chromatograph. Separation, that is different peaks or patterns on the chromatogram, corresponds to different components of the separated mixture. So, you will be getting several components, several peaks. So, different peaks will represent different components of the separated mixture. They represent the different components of the separated mixture, like A, B, C, D and E, which are seen in the figure. You are taking the peak heights corresponds to the concentration of the sample. The heights of the peak A, B, C, D and E are different components, and their magnitude represents the concentration of the component in the sample. Okay, so this is your chromatogram. Coming to the next topic is uses of chromatography. In real life, examples of uses of chromatography are they are used in pharmaceutical company to determine amount of each chemical found in the new product, the amount of each chemical, especially in pharmaceutical companies. Tablets, you will be observing the different components, the composition will be written on them. So, they are used very highly used in pharmaceutical companies. In hospitals, to detect blood or alcohol levels in patients' bloodstream. In law enforcement, to compare a sample found at a crime scene to samples from suspects. Okay, from the crime scene to samples from suspect. Environmental agencies also use chromatography. It determines the level of pollutants in the water supply. Okay, a level of pollutants in water supply. Environmental agencies also use chromatography. Manufacturing plants to purify a chemical needed to make a product. When you are going to make a product in the production or manufacturing plant, you are going to use a chromatograph to purify a chemical needed to make a product. So these are the uses of chromatography. Advantages of chromatography. The advantages of chromatography are the use of longer columns and higher velocity of carrier gas permits the fast separation in a matter of few minutes. Because you have longer columns and the velocity of the carrier gas is very high, so it is a fast separation method which takes place within few minutes. And higher working temperatures up to 5000 degrees centigrade and the possibility of converting any material into volatile component make gas chromatography one of the versatile technique. So you are converting the sample into a volatile component, make gas chromatography. You are converting any material into a volatile component. So this is very versatile technique, and it is popular for environmental monitoring and industrial applications because it is very reliable and can run nearly continuously. It can run nearly continuously. So it is a very reliable process, and it is used in many applications where small volatile molecules are detected and with non-aqueous solutions also. And it is favored for non-polar molecules. It is favored for non-polar molecules. Then what are the limitations of chromatography? Compound to be analyzed should be stable under gas chromatography operations conditions. So the compounds that are to be analyzed, they must be stable under the chromatographic operation conditions, and they should have a vapor pressure significantly greater than 0. The vapor pressure should be greater than 0. The compounds that are analyzed are less than 1000 pascals because it is difficult to vaporize larger compounds, and samples are also required to be salt free. They should not contain ions. Okay, the samples also should be required to be salt free. These are some limitations. They should not contain ions, and very minute amounts of a substance can be measured. Very minute amounts of a substance can be measured, but it is often required that the sample must be measured in comparison to a sample containing the pure suspected substances known as a reference standard. So very minute amount of substance can be measured, but it is often required the sample must be measured in comparison to a sample containing the pure suspected substances known as reference standards. Then applications of gas chromatography. They are used for separation of alcohols and amines. In biomedical field for the analysis of sugars, bile acids and steroids. For analysis of inorganic gases and low molecular weight hydrocarbon gases. It is used to calculate the content of chemical product. For example, in assuring the quality of a product in a chemical industry or measuring toxic substances in soil. Environmental analysis also you are going to use gas chromatography. Then gas chromatography is also used in the analysis of airborne pollutants, performance enhancing drugs in athletics, urine, then oil spills, essential oils in perfume preparations. Okay, to analyze all these you are using gas chromatography. It is very accurate if used properly and can measure picomoles of a substance in a 1 ml liquid sample or parts per billion concentration in gaseous sample. It can even identify, it can be used very accurately if used properly, can measure picomoles, okay, of a substance in 1 ml liquid. And gas chromatography, it is extensively used in forensic sciences, as I told you. In they are used in law enforcement, I told you earlier, that is to compare the sample found at the crime scenes. So they are used in forensic sciences, the disciplines as diverse as solid drug dose, pre-consumption form, identification and quantification, arson investigation, paint chip analysis and toxicology cases employ gas chromatography to identify and quantify various biological specimens and crime scene evidences. So these are the various applications of chromatography. So to summarize the topics which we have discussed today, I have explained to you about absorption. Absorption is a physical or chemical phenomena or a process in which atoms or molecules or ions enter some bulk phase like liquid or solid material. It is a process in which a fluid is dissolved by a liquid or a solute. Fluid is dissolved by a liquid or a solid. That is an absorbent. Adsorption is the adhesion. It is the adhesion of atoms or ions or molecules from a gas or a liquid or dissolved solid to a surface. It is the adhesion of atoms or ions or molecules from the gas or liquid or dissolved solid to a surface. So this process creates a film of the adsorbate on the surface of the adsorbent. Then we have studied about the classification. What is chromatography? Chromatography is a technique for separating mixtures into their components in order to analyze, to identify, to purify and to quantify the mixture of components. Analyzing means to examine the mixture about its components and their relationship to each other. Identifying determines the identity of a mixture or component based on the known components. Purify its separate components in order to isolate one of the interest for the further study. Then quantify determines the amount of mixture and or the components present in the sample. So chromatography is defined as a physical method of separation of compounds of a mixture by distribution between two phases. Okay, by separating the separation of the components of a mixture by distribution between two phases. One is the stationary phase and of larger area, and other is a mobile phase which passes along the stationary phase. Advantages of chromatography over other methods are separation of mixtures of chemicals. Having closer physical or chemical properties can be done easily using chromatography. It is a gentle method, simple and rapid and efficient technique. Separation in even micrograms of the sample is also possible. They are classified in two basic types, that is gas chromatography and liquid chromatography. Gas chromatography is again classified as gas and liquid and gas and solid. Liquid chromatography, it is classified as paper chromatography, column liquid chromatography, column type and thin film type. Then coming to the components of chromatography, the components of gas chromatography are carrier gas supply along with the pressure regulator and flow monitor, sample injection system, the chromatographic column, then thermal compartment or thermostat, the detection system and the strip chart recorder. Gas chromatography is different from other types of chromatography. In this, the solution travels through the column in the gaseous state. So you have the mobile phase which is in the gaseous state. It interacts with the walls of the column. It causes different compounds to elute at different times, called the retention time. The comparison of this retention time is the analytical power of the gas chromatography. So gas chromatography is a type of chromatography in which the mobile phase is a carrier gas. Usually inert gas like helium or an unreactive gas like nitrogen is used, and the stationary phase is a microscopic layer of a liquid or a polymer of a inert solid support. Okay, it has you have a carrier gas, carrier gas. The carrier gas is usually helium or unreactive gas like nitrogen, and you have a stationary phase. It is a microscopic layer of liquid or polymer of inert solid support. Inert solid support. Then the operation of the gas chromatography is normally nitrogen or argon or helium is used as a carrier gas. A variable in compressed form is placed in the cylinder. It is fitted with a pressure regulator. Then it is conducted, the carrier gas is conducted from the cylinder, flows through a flow regulator into the sample injection system, which is maintained at a temperature of T1. Once the carrier gas ensures rapid vaporization but not thermal degradation of the solute gas, and the liquid samples are injected by the syringe. The solute vapor mixes almost instantaneously with the flowing carrier gas, and it is swept into the chromatographic column, which is the heart of the chromatograph. The different solutes in vaporized sample are separated from each other by virtue of their difference in interaction with the column packing. So the column packing is very important here. So depending upon the retention time, in the column, the components, different components have different retention time. They retain themselves in the column, and then they begin to vaporize, or they will begin to elute out, come out of the column. The column is maintained at a temperature T2. It determines the time for passage of the solute. Then the resolution and efficiency obtained with the particular column. At the end of the column, the solutes emerge individually and they enter the detector. The detector produces an electrical signal corresponding to the quantity of the solute leaving the column, and this signal is supplied to the potentiometric recorder. A chromatography is a plot of visual output of the chromatograph. It is we have separation and different peaks or patterns on the chromatograph corresponds to different components of the separated mixture. Each component will be eluted at its own specific time. No two components can have the same elution time. So they will be coming out of the sample, out of the chromatographic column after retention, and they will be entering the detectors. The detectors can be either, their most commonly used detectors are helium ionization detectors. We have nitrogen phosphorus detectors, mass selective detectors, photoionization detectors, and pulse discharge detectors. So the uses of chromatographic column in real life applications, they are used in pharmaceutical company to determine the amount of each compound found in a new product. In hospitals to detect blood or alcohol levels in patients' bloodstream. Law enforcement to compare a sample found at the crime scene to the samples from suspects. Environmental agencies determines the level of pollutants in the water supply. And manufacturing plant to purify a chemical needed to make a product. Advantages of gas chromatography: the use of longer columns and higher velocity of carrier gas permits the fast separation in a matter of few minutes. Higher working temperatures up to 5000 degrees centigrade. The possibility of converting any material into volatile components make gas chromatography one of the most versatile technique. And gas chromatography is popular for environmental monitoring and industrial applications because it is very reliable and can run nearly continuously, and it is used in applications where small volatile molecules are detected, and with non-aqueous solutions also. It is favored for non-polar molecules also, but there are certain limitations. The compound that is to be analyzed must be stable, and it should have a very low vapor pressure, significantly greater than 0 it should have, and compounds analyzed are less than 1000 dynes because it is difficult to vaporize large compounds, and the sample should also be salt free, and they should not contain any ions, and very minute amounts of substances can be measured, but it is often required that the sample must be measured in comparison to a sample containing the pure suspected substances known as reference standard. Then they are used for, in applications, they are used for separation of alcohols and amines, they are used in biomedical field, they are used in analysis of inorganic gases, they are used to calculate the content of chemical products, they are used in analysis of airborne pollutants, and very accurately they are used to measure, they can measure picomoles of substance. They are extensively used in forensic sciences. They are extensively used in forensic sciences. So these are the applications of gas chromatography. So we have understood all the topics, the adsorption and absorption, the chromatography, the classification, and under the classification we have studied about gas chromatography, its components, the construction and working principle, advantages and limitations of gas chromatography, and finally the applications of gas chromatography. So with this I would like to end my presentation today.